In this research, we investigated molecular difference across coronavirus protein spike and NSP1 in the coronavirus phylogeny. Spike enables SARS-CoV-2 to enter the human host cell and begin infection, while NSP1 stalls the host cell's own translation to promote translation of viral proteins. The study explored spike difference in receptor binding and highlighted divergences in host cell penetration and infection. For NSP1, we investigated binding to the host cell 40S ribosomal subunit to highlight potential divergence in host translation inhibition.
We created a flowchart that encompasses all the steps throughout this project, starting with blast searches using distinct databases for proteins and comparing it to SARS-CoV-2. There was a variety of tests performed that can be observed in the chart. We performed a variation of investigations on the phosphorylation site, domain and motif sites, and ribosomal subunit binding sites. Using the data gathered, charts were created for each of the proteins, and statistical testing was performed to create box plots that compared the normality and significance of the genetic variations. We created a phylogenetic tree to map out the evolutionary rate of the proteins. The protein structures for spike and NSP1 were visualized using a program named PyMol.
Phylogenetic trees for NSP1 and spike were divided into 3 clades: MERS, SARS, and outgroup. In both trees, we can see that SARS-CoV-2 belongs to the SARS clade, and it shares the most recent common ancestors with B. cov Rag-G13 and pangolin coronavirus. Predicted motifs were mostly conserved in the spike protein. There was no significant difference from statistical testing involving the SARS clade. We also looked at the conservation of spike experimental phosphorylation sites, which were obtained from the article shown to the right. Statistical testing indicated no significant difference among all clades. Predicted motifs in NSP1 were investigated, showing conservation across all clades. Phosphorylation sites were predicted for NSP1. From statistical testing, we found significant differences between the number of phosphorylation sites only involving the MERS clade. The NSP1 and the ribosomal subunit polar interaction sites were investigated as well.
The proteins were similar and conserved with respect to SARS and MERS. The statistical tests implied differences in structure and function for spike motifs in all clades, and also spike's phosphorylation sites had significant differences with respect to all clades, suggesting they're changing in a clade-specific manner. Tests also confirmed conservation of structure and function for SARS-2 NSP1's phosphorylation sites with respect to the other clades. The polar interaction sites between protein and subunit showed no motif sites involved in binding to the ribosome, but one phosphorylation site is. Phosphorylation also appears to affect NSP1's interaction with the ribosome and could play a role in regulating viral protein replication. Conservation makes both proteins viable targets, and phosphorylation is a feature that may play a larger role in the function of both proteins. Further investigation can yield insights into possible drug target sites to treat COVID-19.